So today we'll talk about the role of rootstock and its genetic background in plant mineral status. So thank you for the scientific committee, because the recognition is for me very important, and as a young researcher, it's very important to be promoted and to be recognized for our work. And I think I can say on the behalf of the author that for us it was very important to publish in an open access journal, and also the transfer from the scientists to the private. That was also very important because we didn't IVES technical reviews. So I'm working on rootstock cyan interaction. So it's a CIF under agreement PhD thesis. So I'm working with NSC company and the laboratory of a GAV in Bordeaux. So this work is included in the biggest program, that is the PG program, that is launched in 2023 by NSC. And the idea is to support the process of evaluating and understanding rootstock adapting to climate change. So what we are doing in this program, we characterize existing and also newly created rootstock at a GAV, but also in the Conservatoire of Vignoble Charentais, headed by Sébastien Julliard. So what we are doing, so this characterization is done on GrefAdapt, so what Case presented a bit before in Bordeaux, but also in Charente. So what are the criteria for this program? So we evaluate many different agronomic traits of interest, such as drought tolerance, but also in the context of agroecology, where we want to reduce the input in the vineyard. So it's interesting to have a rootstock that are more tolerant to mineral deficiencies. So the final goal, the final goal of this work is to suggest 5 to 10 new rootstock. So this PGDFI program is included in the biggest program, that is the rootstock improvement program, led by UMR GAV and coordinated by Nathalie Ola. So in this program, we have 3 stages. So the first one is to phenotype and multiplicate stage, where we have new hybrids and new mapping population, and we studied different agronomic traits, and the idea is to identify the genetic architecture of drought or confer vigor or tolerance to calcareous soil, so of the new crosses. We have the intermediate selection stage with GrefAdapt plot. In this case, we use existing rootstock. And finally, the idea is from these 2 first stages is to have 5 to 10 rootstock to the final stages, at the stages where we are testing in multi-site this rootstock. And above all this pyramid of improvement program, we have this information system. So it's a huge database where we collecting information about rootstock. So a bit like what Gonzaga presented this morning for Spain, we have the same system in France. And this huge database, we have a work ongoing that is coordinated by IVES and Laurent Deguin. And so they are working on this in the context of PNDV. So let's talk about rootstock. So mineral disorders, and we know that we have a negative impact on yield, but also on growth-related traits. And our rootstock, that is the hidden part of the vine, is the hydromineral pump, and is known to have an impact on the whole plant, and especially on the cyan, and it can also modify the whole plant development. There were some first observation in 1956, so very old observation, that we already observed impact of the rootstock on deficiency symptoms. But the large diversity of rootstock is poorly studied. So our question is what the rootstock effect on plant mineral status, and also is it related to the observation of mineral deficiency observation? So this work was carried out on GrefAdapt. So this is the vineyard, experimental vineyard. So it's planting. Okay, thank you. So this plot was carried on GrefAdapt. So it was planted in Bordeaux in 2015. we are in the sandy gravy soil, and the management is border management, so we have about 6,000 vines per hectare in the Guillaume mixed training system. And we have free bracts because we have a bit of heterogeneity in soil composition. So what about plant material? So on Greffe Adapt, we have 5 scions, which are Cabernet Sauvignon, Grenache, Syrah, Pinot Noir, and Uni Blanc. And these 5 scions represent the main area of wine growing in France. Also, they are very different in terms of genetics. And what is very interesting is that we have 55 rootstock. So we have 30 French rootstock, 30 rootstock that are allowed in France to be planted and used is the list of the plant. So we have only 30 because the last one is sensitive to phylloxera. So there is many well-known like 3309 or 1110 Richter, SO4, Fercal, and so on. But we have also 25 foreigner rootstock that are rootstock reported by the literature as interesting in context of climate change because drought tolerant or calcareous tolerance. So we have, for example, the Milano series. They are pretty young on this experimental design, but we have this one. We have also the Georgicon series and other like Binova or Borner, Ramsey, Schwartzman, and so on. So finally, we have 55 rootstock grafted on 5 scions, and we have 275 combinations and 50 plants per combination. So it's a lot of plants. So we can work on each genotype, but the idea was also to understand the effect of the genetic background. So for that, what we decided to do is to group genotypes per genetic background. And we assigned the genetic background when we had at least 55% of the genetic background present. So instead of working with 55 genotypes, we also work with 7 different genetic backgrounds. So what we did, so we sampled petiole 2 years, so in 2020 and 2021. So the plants were 5 and 6 years old, respectively. We did this sample at Veraison. So we did 6 replicate per combination. So it's about 1,300 samples per year. And we analyzed 13 elements. So we have 7 macroelements and 6 microelements. But after, we'll focus more on the microelements. So we also assess many traits, so growthology traits on this on Greffe Adapt. Every year we assess pruning weight, conserve vigor, dual X data to have a proxy of nitrogen, for example, but also the shoots, the number of shoots per vine. And we also have yield-related traits, such as the bunches number or the fertility, which is the number of bunches per shoot. And finally, in 2021, what we observe is magnesium deficiency symptoms. So this magnesium deficiency symptom is due to a disequilibrium of the potassium-magnesium ratio. And we decided to make a scale, so from 0 to 3, from 0 to 3. So 0, we had no symptom of magnesium deficiency. 1, just the first bottom leaves. 2, that is half of the canopy were expressing magnesium deficiency. And finally, the highest number is when all the canopy was expressing magnesium deficiency. So what are the results of this? So what we show is that we have a strong and a stable effect of the rootstock when we study the 55 genotypes. So for main macroelements, so magnesium, sulfur, phosphorus, or potassium, we have the rootstock that is dominant or equivalent to the scion, except for nitrogen, that scion is dominant. But what's very interesting to see that the rootstock is stable. The effect of the rootstock is stable across 2 years, the 2 years of the study. His dominant factor on macroelement content of petiole except for nitrogen. And the rootstock explained from 7 to 45 of the variance of mineral content. So this is the first result on the 55 genotypes. But when we look at the genetic background, so in this case, so we look at the effect of the 7 genetic backgrounds on mineral content. So we define state 2 based on the values. And what we observe is that we have, first, a significant effect of the genetic background on plant status, that many several genetic backgrounds have stable effect. So we find exactly the same result from 1 year to another year. And for example, Vitis riparia had a higher proportion of plants in magnesium or phosphorus deficiency. But that's very interesting is that Gauthier had in 2020 showed this result on 1 year plants. And in this case, we are able to find the same result but on 5 and 6 year plants in the field. So we have very strong and robust effect of genetic backgrounds on some elements. But it's not so easy when we try to link the science development with the mineral analysis. So we didn't find a lot of relationship between mineral and all the information about growth-related trait or yield-related trait, except for potassium or boron, but I represent potassium. So for yield-related trait, when we look at potassium, even though we are in optimum, above the optimum, because the optimum is 1.5, you can see that we are a relationship between the potassium and the number of bunches. So the higher the potassium is, the higher the number of bunches is. And it's also true for growth-related traits, such as pruning weight. We have a strong relationship between potassium content and pruning weight. So we have this positive correlation between potassium and growth and yield-related trait, but it's not easy to find relationship between mineral content and growth or yield-related trait. It can be explained because of the vineyard is well supplied, but we have effect on rootstock on mineral content, but it's not so easy to link with the behavior and the phenotypic behavior of plants. But now we will look at the mineral deficiency observation. So this is the scale we did. So this is the classification of all our rootstock, so the 55 rootstock according to the deficiency symptom. And if you can look at, I don't know if it's easy, but the rootstock with the highest number of, the strongest magnesium deficiency is 4453 Maleg. So it's pretty well known that this one is magnesium deficient more than other rootstock. So, okay, we are not surprised that rootstock behave as we were expecting. We are also able, for the first time, to characterize new rootstock like geological series, like the 121 or 251, that we have no strong magnesium deficiency. And what is interesting is that with the panel of 55 rootstock, finally we have a diversity of responses to magnesium deficiency symptoms. This is what we can see. If we link this, so this is the same classification, but if we look at the mineral content of the petiole. So globally, it's working, and the threshold we use in literature are suitable for most of the rootstock. But if we go into detail, we can see some difference. So this is the severity of the deficiency symptom observed, and we characterize if in the petiole they were deficient, optimum, or excess. So when we have no symptom, okay, we have less deficient petiole. So it's pretty normal. And we have higher number of individuals that are optimum and excess. And when we begin to have symptoms, when we look at the petiole, so we have highest number of individuals with deficiency. But as you can see, it's not because we have no symptom that petiole analysis say everything is fine. We have some rootstock that are deficient if we just look at the petiole. It's the case, for example, of Viala. That is a rootstock. If we look at the petiole analysis, we can say, oh, wow, it's not going well. He has 85% of the plants in deficiency. But finally, if we go in the vineyard and we look at the plant, everything is fine because it has like less than 20% of the plant in deficiency with symptom. So what is interesting is the threshold we use in literature about 0.5, 0.6 is suitable for most of the rootstock. But finally, we have some difference of satiety depending on the rootstock and the cyan interaction. So to conclude, if I have some key messages, is that the rootstock effect is a dominant factor except for nitrogen. We have significant effect of genetic backgrounds on mineral status. And finally, fertilizer management must take into account the rootstock-cyan combination. This perspective of this work is to characterize also the mineral status at the adult stage, because the experimental vineyard will be 10 years old, so it will be interesting to see if we have the same tendency or not, and to suggest an exhaustive classification of rootstock magnesium behavior. So thank you for your attention. I want to thank IVES annual meeting organization for all this work and this amazing week. The IVES scientific committee, again, because of this very important as a young researcher to be promoted. And my co-author, especially the AGV team, but also NSC and Mathilde and Xavier for their support. Thank you.